Good afternoon, everyone. For those of you who don't know me, I'm Stephen Burley. I'm the director of the Institute for Quantitative Biomedicine and the director of the RCSB Protein Data Bank, and I'm delighted to welcome you here to the second of our fall 2023 seminar series. Before I introduce today's speaker, I wanted to, I'm having difficulties. Okay. Before I introduce today's speaker, I want to just draw your attention to the schedule for the remainder of the semester, and thank Judith Penzes from our community here from IQB, and also Anders Larsen, who will be giving the last talk of the season. Another congratulations to Jennifer Jiang, who was awarded the 2023 IQB Inclusive Leadership Award in recognition of outstanding achievements. We are keen to get nominations for the 2024 IQB Inclusive Leadership Award, and please see Michelle Sangera for details or our website, and either nominate yourself or nominate another individual who you think would be highly deserving of the honor, and reflect the tradition that we're continuing with Jennifer. We've got a number of events coming up being run by the RCSB Protein Data Bank that I wanted to just draw your attention to. The first is a set of presentations and workshop on leveraging the APIs, the application programming interfaces that underpin all of the RCSB PDB activities, including all of the rcsb.org. So this is intended for individuals who are interested in performing both bioinformatics analyses and going on to machine learning. There'll be an introduction to the APIs initially, and they were running two hands-on exercises for different time zones, one focused on this side of the world, and another one timed to make it easier for people on the opposite side of the world to attend in Asia. So that's going to take place on the 12th and the 19th of October. Also on the 19th, not conflicting by virtue of different participation demographics, will be a workshop that we're putting on jointly with ASBMB, the American Society for Biochemistry and Molecular Biology, and I believe we have the president of ASBMB in the room. Is that correct? Ann Stock is this year's president of ASBMB. Thank you for your service. So the focus for this ASBMB workshop, again, joint with RCSB PDB, will be teaching enzymology with the Protein Data Bank from the pandemic to Paxlovid. And I know that there's at least one person in the room who's benefited from Paxlovid for acute treatment of SARS-CoV-2 infection. So please put this in your calendar if you're interested in participating, and register. It's free, but you do have to register. Same, excuse me, for the API workshops on the 12th and the 19th. And then the final event I want to publicize is our 2024 Winter Boot Camp on Science Communication that will be repeating. This the applications are due by email on the 31st of October. You can find the details on our iqb.rutgers.edu website. This is an opportunity for undergraduates and graduate students to work intensively together to develop expertise in both writing and molecular visualization. The work product of this activity will be published on pdb101.rcsb.org, our training, outreach, and education web portal that complements what we're doing with our research-focused web portal, rcsb.org. And then finally, we have a number of job openings both here at Rutgers and at UCSD, where we have another big RCSB PDB team, and the opportunities are listed here. please go to our jobs page, www.rcsp.org/pages/jobs for more information. So with that, I will stop, and I'm going to hand over to our speaker today, Squire Booker, who did his PhD at MIT with Joanne Stubby, and then went on to postdoctoral work in Paris. Very nice place to be a postdoc. Yeah. And has been at Penn State for many years, where he's risen through the ranks, received numerous awards and appropriate recognition given his the work that he's been doing in crystallographic enzymology. He's a Howard Hughes Medical Institute investigator, and he's also a member of the National Academy of Sciences. So, Michael, I'll let you make the transition. Welcome, Squire. It's great to be able to host you here.
So thank you for the kind invitation to come here. It's been amazing. I always enjoy. I'm a big football fan, right? And I always enjoy going to other Big Ten schools. And so this is my first time at Rutgers. But I've had an amazing, I've only had 2 meetings so far this morning outside of meeting with Dr. Burley, but they were both amazing. Met with the chemistry, head of the chemistry department, and I met with the PDB team, which was particularly enlightening. I always wondered what happens when we submit these structures for validation. And so they took me through, the whole process to tell me what they do and the kind of information I get out of the PDB. Okay. And so today I'm going to talk to you about, well, I'm going to start off telling you about our studies on biological methylation, okay? And I just need to get my slides to change. This happens quite often, whereas the first slide, I may need some help. Well, it's stuck. But that happened last week while I was at another university. So I think you did the right thing. I think I needed to press the space bar.
Yep. Awesome.
Okay, thank you so much.
Oh, sorry, I think it's just on.
Okay. So for a number of years, my laboratory has been interested in biological methylation. And on this particular slide, I'm showing you the structure of S-adenosylmethionine, which is the primary methyl donor in the cell. For the chemists in the audience, this is the cell's version of diazomethane, right? And S-adenosylmethionine can be used to methylate proteins, carbohydrates, lipids, DNA, RNA, and a variety of small molecules. And the reaction for biological methylation is actually quite straightforward. All you need to do is generate a nucleophile, and then the nucleophile attacks the activated methyl group of S-adenosylmethionine. We know something about the transition state of the reaction from isotope effects and other similar studies. That drops down to give you your methylated product and this co-product, S-adenosyl homocysteine. And using that mechanism, we can methylate a variety of different atoms. We can methylate nitrogens, oxygens, sulfur, selenium, arsenic, and tellurium. And we can even methylate carbon, but only if we can generate a suitably nucleophilic carbanion, right? However, for over 40, maybe 50 years, we've known that in nature, there are a variety of molecules that receive methyl groups on carbon atoms that are not activated. In other words, you can't remove a proton from them to give a carbanion because the pKa values are too high. And here are just an examples of a few of these molecules. In some cases, you can methylate an sp2 hybridized carbon. And sometimes you can methylate an sp3 hybridized carbon, and you can also methylate a phosphonate phosphorus atom. And what we know today is that all enzymes that catalyze these types of methylation reactions use radical strategies to do so. And all the enzymes that do these reactions belong in a superfamily called radical S-adenosylmethionine. And all of the enzymes in this superfamily have a 4-iron 4-sulfur cluster cofactor, shown right here, wherein 3 irons in that cubane cluster are connected to the protein by 3 cysteine ligands. That leaves 1 iron in that cluster with an available coordination site to which S-adenosylmethionine associates, as shown here. Now, many of us know that clusters are redox active. They can gain and receive electrons. When these clusters receive an electron from an external donor, that causes a scission between this carbon and sulfur bond to generate methionine, which is bound to the cluster, and this potent oxidant called a 5' deoxyadenosyl 5' radical, all right? And the role of this oxidant is to initiate some sort of catalytic event, typically by abstracting a hydrogen atom from an enzyme-bound substrate. And so we know that there are about 700,000 individual sequences of these radical SAM enzymes, and they catalyze well over 100 different reaction types. You can identify a radical SAM enzyme typically fairly easily because they have a cis XXX cis XX cis consensus sequence for those cysteines that ligate to the iron sulfur cluster, okay? Now, of these greater than 100 different reaction types, biological methylation is one. And these reactions have been divided into 4 classes: Class A, Class B, Class C, and Class D. The Class A enzymes use a cysteine dyad to methylate these amidine carbons on RNA substrates to give a methyl group at C8 or a methyl group at C2. The Class B enzymes tend to be more versatile because they can methylate sp2 hybridized carbons as well as sp3 hybridized carbons in phosphonate phosphorus centers. And these enzymes require this methylcobalamin cofactor in catalysis. So that's one of the reasons why this morning I kept asking the PDB people, can we bring up some structures with methylcobalamin bound, right? And then the Class C enzymes right here use 2 molecules of S-adenosylmethionine simultaneously bound to catalyze methylation, methylene transfer, or cyclopropionation only on sp2 hybridized centers. And then finally, there's this Class D family of radical SAM enzymes, which is represented by an enzyme called MJ0619. The MJ comes from methanococcus genashii, and it was purported that this enzyme methylates 2 carbons on pterin to generate methanopterin. All right, so methanopterin is a C1 carrier in methanogenic archaea. And again, MJ0619 was believed to add methyl groups to this carbon and that carbon to generate methanopterin. And so in the Class A, B, and C enzymes, S-adenosylmethionine plays 2 roles. It's the source of the radical, the 5' deoxyadenosyl radical that I showed you. And it's also the source of the methyl group that's appended in the reaction. In the Class D enzymes, S-adenosylmethionine is the source of the 5' deoxyadenosyl radical, but the previous authors who had studied this suggested that the source of the methyl group was methylene tetrahydrofolate rather than S-adenosylmethionine. And because of these differences, we decided to study these class D enzymes since we had already studied extensively class A, class B, and class C enzymes. So here's the molecule, methanolterin, that we're going to talk about, all right? And it looks a lot like folate, except for differences in the tails, as you can see at the top and the bottom. The tail in methanolterin is more extensive than the tail in folate. The differences in the oxidation state of this particular carbon. At the top, we have a methylene, and then at the bottom, right here, we have a carbonyl. And then, of course, the main differences is that methanolterin has methyl groups at C7 and C9, whereas folate does not have methyl groups on those positions. Okay, so the previous people who studied this reaction showed that if they expressed the gene for MJ0619 heterologously in E. coli, they could isolate methylated pterins, suggesting that E. coli has what you need to do this reaction. When they used labeled methionine, they showed that the label did not show up in the product, meaning that the methyl group does not come from S-adenosyl methionine, since methionine is the precursor to the methyl moiety in S-adenosyl methionine. And then studies with label acetate suggested to them that methylene tetrahydrofolate is the methyl donor. And so here's part of the primary structure of MJ0619. And the first thing that you'll notice about this part of the structure is that there are two of these cis XXX, cis XX, cis motifs, one towards the extreme N-terminus and one a little farther down in the sequence. And based on this information, or based on this sequence, the authors suggested that this enzyme might have two 4-iron 4-sulfur clusters to which S-adenosyl methionine associates. And they hypothesized that the substrate is most likely pterin, because pterin is found both in E. coli and in Methanocaldococcus genacei. And we know that Methanocaldococcus, that MJ0619 can produce methylated pterins. So they suggested that one SAM molecule binds to the 4-iron 4-sulfur cluster ligated by these cysteines. It's cleaved to generate a radical that leads to methylation at one position, shown right here. And the second SAM molecule binds to a 4-iron 4-sulfur cluster ligated by these cysteines. It's cleaved to a radical to install a methyl group at this particular position. And again, it was postulated that methylene tetrahydrofolate serves as the methyl donor in the reaction rather than S-adenosyl methionine, all right? And what I can say is that in 2 years of our own studies, we were unable to corroborate any of the findings that I explained to you, okay? And then the pandemic hit. And right before the pandemic hit, Cody Lloyd, a graduate student in the lab, he's the one with the hat, was able to isolate this protein, crystallize it, and then do X-ray diffraction studies on it. So we did remote data collection. And we were able to, and during the pandemic, he used his time wisely while we were all shut out of the labs to learn how to fit electron density maps. So he learned X-ray crystallography. And he determined the structure of this protein to 1.85 angstroms resolution, showing that it consisted of 4 domains. There's this N-terminal domain up here from left to right, which is small, which is followed by another small domain that we term the N-terminal auxiliary cluster domain, which is followed by this radical SAM core domain, shown right here, okay, which is then followed by a domain that we call the C-terminal auxiliary cluster domain, all right? Now, what I'm gonna do is bring up the structure of the radical SAM core, all right? So this is sort of the core of the protein. It consists of a beta 6 alpha 6 partial triose phosphate isomerase barrel, okay? Typical TIM barrels are beta alpha 8, right? And they form a true barrel, but beta alpha 6 sort of gives you like a curved fence, all right? This domain of the protein binds the iron sulfur cluster to which S-adenosylmethionine associates. This is the domain where the radical is created to initiate catalysis. Now, in all of our structures, instead of S-adenosylmethionine, we show the cleavage products of that reaction, which are 5'-deoxyadenosine, get this cursor to work, shown right here, and methionine. And we do our crystallizations in the presence of these products for a reason that will become apparent in just a few minutes, all right? Now, what I'm gonna do is I'm gonna turn this about 90 degrees so then I can build up the rest of the structure on top of it, okay? The second domain that I wanna talk about is the C-terminal auxiliary cluster domain. It's the largest domain in the protein, being composed of about 239 residues. And it serves to sort of close off that open fence on the radical SAM core and to create the active site for the protein, all right? This domain also has a 4-iron 4-sulfur cluster associated with it. Now, just upstream in sequence of the radical SAM core domain is this N-terminal auxiliary cluster domain, which I show in tan. It's small, only 45 residues, but it also houses a 4-iron 4-sulfur cluster, all right? And then the last domain at the extreme N-terminus is this rubridoxin-like domain shown here in pink, which I think is really beautiful looking, all right? Now, interestingly, all of these metallocenters that I'm showing you are in atypical ligation spheres, all right? We've already discussed the radical SAM 4-iron 4-sulfur clusters. Historically, iron sulfur clusters have been thought to be ligated by 4 cysteine residues. In this particular case, radical SAM proteins only have 3 cysteines that are ligated to the cluster, and that allows S-adenosylmethionine to ligate to that 4th iron in order to do the radical chemistry. The N-terminal domain has 3 cysteines and a histidine ligand. Now, that has been seen before, but it's very rare to have 1 histidine ligand in a 4-iron 4-sulfur cluster. The rubridoxin-like domain has 3 cysteines and a histidine ligand, whereas almost all other rubridoxins have 4 cysteine ligands. And then lastly, there's this C-terminal domain, which has 3 cysteines and a methionine ligand. That had never been seen before, to have methionine as a ligand to an iron sulfur cluster. And it appears that the ligand is relatively weak because if we do our crystallizations in the presence of thiocyanate, it displaces the methionine and it itself becomes a ligand, okay? Now, as I said earlier, we were unable to corroborate the findings of others who had worked on this protein before we did. And so we were left with this question. We have this structure, but we have no idea what this protein actually does, all right? Now, if we looked closely at the active side of the protein, we saw electron density that we could model, right, as 2 molecules of phosphatidic acid. Here's the head group of 1 molecule shown right here. Here's the head group of another molecule shown right here. Here's 1 lipid chain going back right here. Here's a second lipid chain, going back here. And on the other phospholipid, here's 1 lipid chain. And there's the second lipid chain. And so this is what phosphatidic acid would actually look like. But given that the electron density wasn't that robust, we decided to look for another method that we could use to verify that phospholipids were indeed bound in this particular protein. And so we turned to a mass spectrometry-based method, high resolution native spray mass spectrometry. And so in this case, what we're going to do is ionize the protein under very gentle conditions so that it maintains its cofactors and substrates intact on the protein. And so I'm going to show you 3 regions of the chromatogram. Here's region 1, region 2, and region 3. Region 1 would correspond to proteins that have no lipids bound. Region 2 corresponds to proteins that have 1 phospholipid bound per protein. And region 3 corresponds to proteins that have 2 phospholipids bound per protein, as we had seen in our structure. All right. Now, when we do the ionization, either because of oxygen that we let in to the reaction accidentally, these iron sulfur clusters can't tolerate very much oxygen, or because of the oxidation process, we get a number of different breakdown products of the iron sulfur cluster, which we can actually see by mass spec. Now, before we do the experiment, we use a technique called Mossbauer spectroscopy that allows us to look at what the iron actually looks like in solution. And Mossbauer tells us that all of our proteins have 3 4-iron-4 sulfur cluster and 1 mononuclear iron, which is in the rubidoxine site. But during the ionization process by mass spec, we can see all of these different breakdown products of the cluster. And so these letters above each of these peaks correspond to the type of breakdown product that we see. P, for example, corresponds to a protein that is fully reconstituted. So 3 4-iron-4 sulfur clusters and a mononuclear iron. So in region 2 here, we have P, and then we have N, which corresponds to 3 4-iron-4 sulfur clusters, but we've lost the mononuclear iron, okay? Importantly, if you look above each letter, there's a symbol, and the color of the symbol tells us what sort of lipid we see in the active site of the protein. And because we're using such a high resolution mass spectrometry method, we can determine exactly what lipids are present. So for example, in this particular species where there's N, you go up here and you see that that corresponds to a protein with phosphatidylethanolamine in it. One lipid chain has 16 carbons with no double bonds, and the second lipid chain has 17 carbons, which is sort of weird, right? Because lipids are built 2 carbons at a time. So most lipid chains are even numbered, but we know that as E. coli enters stationary phase, it will cyclopropionate double bonds on some of their lipids. And so that's where you get 17 carbons. 17, the C stands for cyclopropyl group, all right? So we see that on the protein. And then we can go to species, for example, like here. Here's P again with black and yellow. Black corresponds to 16 carbons, no double bonds, 17 carbons with a cyclopropane ring, and yellow corresponds to 16 carbons, 1 double bond, and then 18 carbons with no double bonds. So based on our high resolution mass spectrometry in concert with our X-ray crystal structure, we know that this protein is capable of binding E. coli phospholipids, all right? But now we have a bit of a problem because Methanolcaldicoccus genashii is an archaean. Its membrane lipids look quite different from bacterial or eukaryotic membrane lipids, all right? So on the left here, I'm showing you what archaeal membrane lipids look like. They're synthesized via the isoprenoid biosynthetic pathway, and that's why you see these methyl projections on the structure. And on the left side, I'm showing you a picture of bacterial or eukaryotic membrane lipids. Now, as I said, archaeal lipids are isoprenoid based. That's why you see the methyl projections. By contrast, bacterial and eukaryotic lipids are fatty acid based, all right? Archaeal lipids are ether linked. So you see these ether bonds here, which make them incredibly stable. And bacterial and eukaryotic lipids are ester linked. So you see ester bonds there. Archaeolipids contain a glycerol 1-phosphate backbone, whereas bacterial and eukaryotic lipids contain a glycerol 3-phosphate backbone. And that gives you an inversion of stereochemistry at C2 between archaeolipids and bacterial lipids. And so these lipids really are quite different. And so the question is, if we want to study this enzyme, if we want to try to ascertain what it actually does, we need a source of archaeolipids. And so where are we going to get archaeolipids? Well, let us telephone Bill Metcalf, all right? And Bill Metcalf is an incredible microbiologist at UIUC, University of Illinois, Urbana-Champaign. And he's an expert on this organism called Methanosarcina acetovorans, which is another archaeon. Our protein is from Methanocaldicoccus genacei. But Bill even has a genetic system in acetovorans. And so we were hoping that he could give us a very sophisticated answer to the problem that we had associated with lipid substrates. And what he told us is that, well, we isolate acetovorans all the time. And what we'll do is take what we want every time we grow the organism up, and then the rest we'll just spin the pellet down and send it to you guys. And so he said, we're happy to send you our waste, all right? And so we took Bill's waste, which was generated by his postdoc, Matteo Casu, and we extracted the lipids from the crude lysate pellet and mixed it with our protein, MJ0619. And then we heated that mixture up at 40 degrees Celsius to allow the bacterial lipids to exchange out, and then the archaeolipids to exchange in. And then we repurified the protein over a gel filtration column, PD10. And then we repeated that same experiment that I told you previously, the native spray mass spectrometry experiment, which I won't go into detail on this particular slide. But again, we see 3 regions of the chromatogram, with region 1 having no lipids, region 2 having 1 lipids per protein, and region 3 having 2 lipids per protein. And the important point is, if you look at the symbols above each of these letters, they correspond to lipids that are found, pretty much only in Methanosarcina acetovorans. So we'll see, for example, we have here archaeotidyl inositol. Over here, we'll have archaeotidyl serine. And the interesting thing is that acetovorans will hydroxylate certain positions on their lipids, and we actually see those hydroxyl groups on the lipids by high resolution mass spectrometry. Okay. So we took this protein, which had then been reconstituted with these archaeolipids, and redetermined the X-ray crystal structure in the presence of archaeolipids. We got better electron density. And when we looked deeply into the active side of the protein, we noticed something very interesting. So here you see 2 lipids. One is shown right here in orange, with one tail going in this particular direction, and the second tail going in this particular direction. And then the second lipid is shown here in yellow, with one tail going in this direction and one tail going in that particular direction. But strikingly, this tail in this particular lipid terminates only a few angstroms, like 3 angstroms away from the 5' carbon of 5' deoxyadenosine. Now, if you remember, I told you that when you generate the 5' deoxyadenylyl radical, its job is to initiate catalysis by abstracting a hydrogen atom from the substrate, and that gives you 5' deoxyadenosine. So the reason why we do our structures in the presence of methionine and 5' deoxyadenosine is where that 5' carbon of 5' deoxyadenosine is, in relationship to where the substrate is tells you where the site of chemistry should be. And so this picture here is telling us that this enzyme is probably doing something at the extreme end of the lipid tail, all right? At that point, we had no idea what the reaction was, but we went to the literature. We said, what reaction in archaeal lipid biosynthesis is not known, all right? And we stumbled upon a protein called glycerol dibiphytanyl glycerol tetraether synthase. And this was a protein that people had been looking for about 40 or 50 years. And the reason why is this. In some archaea, they will take lipids at opposite sides of the membrane and make carbon-carbon bonds between the very ends of the lipids to give this macrocycle, which is called glycerol dibiphytanyl glycerol tetraether, or GDGT, all right? And this is sort of a cartoon of what these lipids look like in a membrane. So the black would correspond to your normal membrane lipids, and the red would correspond to your GDGT lipids. And so you see this macrocycle spans the membrane, and that gives the membrane rigidity. It enables archaea to exist at high temperatures because these macrocycles control the fluidity of the membrane. It gives them the ability to tolerate extreme pH values and extreme salinity. And in fact, the molecule is so stable that paleoclimatologists use GDGT as a biomarker to reconstruct past temperature cycles on Earth. And the reason why is because it's typically made in archaea when the temperature gets really high. And so the paleoclimatologists can then look at different layers of the Earth's crust and see if GDGT is present. If so, that tells them something about the temperature on Earth at that particular time, all right? Now, people had been looking for this enzyme for 40 or 50 years and were unable to find it, but they had hypotheses for how an enzyme might construct a carbon-carbon, construct these carbon-carbon bonds to generate, this macrocycle. So the biosynthetic pathway starts with the isoprenoid biosynthesis, which starts with isopentenyl diphosphate and dimethyl diphosphate, and you take those and you build up this particular molecule. And then there's an enzyme, a flavin-containing enzyme called geranyl-geranyl reductase that will reduce all of these double bonds to give you this molecule called archaeol, which is a common constituent of archaeal membranes. And then from archaeol, you might cyclize directly to give you GDGT. The problem is that from a chemical standpoint, it is very challenging to tether two completely unactivated sp3 bonds. And in biology and nature, that reaction was completely unknown, okay? A second possible pathway is that you get to this particular intermediate that I showed you previously, but geranyl-geranyl reductase might only partially reduce it. And then from this structure, you could do macrocyclization. And that advantage is that at least you have some functional groups that might make the macrocyclization easier to do, okay? And so we decided to test this controversy in the field. We took lipids that we extracted from imacetylvorins and mixed them with MJ0619. We exchanged out the bacterial lipids, exchanged in the imacetylvorins lipids, and then again, we purified the protein over a PD10 column. And then there were 4 lipids that we used as markers. The top left, I'm showing you archaeotidyl inositol that has double bonds in lipid chains. I'm showing you archaeotidyl glycerol that has double bonds in the lipid chains. And then at the bottom, I'm showing you archaeotidyl inositol that has no double bonds in the lipid chains. and archietyl that has no double bonds in the lipid chains. And so we're using those as markers for this particular reaction. And so as you can see in this top slide here, if we look at the molecules that have double bonds over the course of a 30-minute reaction, we see that those don't seem to decay in concentration. By contrast, if we look at the molecules at the bottom that don't have double bonds, we see that both of these molecules decay, and we also see the formation of GDGT. So this tells us that this enzyme is using a completely saturated lipid as the substrate to make these carbon-carbon bonds to give these macrocycles. And so a research scientist in the lab, Bo Wong, who's covered up by the Zoom picture, was able to synthesize a pure substrate for our reactions, which I showed down at the bottom right. Enzymologists love pure substrates, right? And then we use the pure substrate in our reactions. And so, as you can see, if we take the archylglycerol synthesized by Bo, we show that that substrate can then give us GDGT, which is this huge macrocycle shown right here, all right? We also see the production of a small amount of GTGT, which is sort of a semi-macrocycle where you've made one carbon-carbon bond, but you haven't put the second carbon-carbon bond together to make the fully matured macrocycle, okay? Now, I want you to take a look at the y-axis on this particular slide. It doesn't appear that we're making a lot of this particular molecule, but at the same time, we don't have standards synthesized that will allow us to quantify exactly how much of this molecule is being made. Now, unexpectedly, we saw a second molecule being made from our archylglycerol substrate. And that molecule is called archylglycerol macrocycle, where you tether the two ends of the same lipid together to generate this smaller macrocycle. And based on the response in the mass spec, it appears that the smaller macrocycle is made at a higher concentration than the larger macrocycle, okay? And so this led us to then conclude that the role of MG0619 is this elusive GDGT synthase that people had been looking for. And so I just want to say that Paula Waylander's laboratory at Stanford and a collaborator in China, Jiri Zhang, also identified a GDGT synthase using complementation type procedures. And their paper was published a few months before our paper was published. Now, we went on to do some mechanistic studies, okay? And so our studies showed that GDGT synthase, or MJ0619, can take archylglycerol and make archylglycerol macrocycles, shown right here, as well as GDGT. So it's clear that we can make carbon-carbon bonds from CSP3 completely unactivated precursors, which had never been seen before in biology. In fact, in biology, you typically make carbon-carbon bonds via polar mechanisms, right? Here's a classic case, for example, where you'll have, a carbonyl. The protons adjacent to the carbonyl can be made acidic. You remove a proton to generate a carbanion, shown up here, and then that carbanion can attack an electron-deficient carbonyl to give you your carbon-carbon bond. So in biology, that's largely the way that carbon-carbon bonds are made, right? But here we know, based on these molecules right here, that we can't remove any protons because the pKa values of those lipids are 50 or above, right? So we can't do acid-base chemistry on these particular substrates. So I did a second postdoc at the University of Wisconsin at Madison, where I worked for Perry Fry, and he published this very influential textbook called Enzymatic Reaction Mechanisms in 2017. And a passage in this particular textbook says, in chemistry, many methods are available to synthesize carbon-carbon bonds, and the reactions proceed by both polar and radical mechanisms, right? However, he says, enzymatic ligation of two molecules through carbon-carbon bond formation invariably proceeds by a polar mechanism. And at that time, in 2007, that was actually the truth. But because of the radical SAM superfamily and our learning of the breadth of the reactions that they've catalyzed, we've started to learn that nature can use radicals to make carbon-carbon bonds as well. And one of the first places is this enzyme called benzyl succinate synthase. It's one of these glycyl radical enzymes. It has a stable glycyl radical on its polypeptide backbone. The glycyl radical is generated by a cognate radical SAM activase. And so the glycyl radical is then used to generate a cysteine radical on the protein, which abstracts a hydrogen atom from the substrate, which in this case is toluene. And that gives you this toluene radical that I show up at the top left. And then that toluene radical can add into this double bond, this pi system, to give this product radical. And then the product radical can then grab a hydrogen atom from that cysteine that I told you about, all right? So that had become known by the time that we studied MJ0619. The problem is that there are no double bonds in our substrates, right? So we don't even have that. All right, so here's our working hypothesis. Here's our challenge. Here's the conundrum that we have, all right? So our working hypothesis is we're going to use one molecule of SAM to generate a radical that abstracts a hydrogen atom from the tail of the lipid, and that gives this carbon-centered substrate radical. Then we're going to use a second molecule of SAM to abstract a hydrogen atom from this lipid to give a second substrate radical on a different lipid. And then these two radicals will come together, will recombine to generate your carbon-carbon bond. That's how we think this reaction should take place, right? The problem, however, is these radicals are incredibly unstable. So how do we stabilize that first high energy radical that we generate up here while we're generating this second radical down here, okay? How do we do that? So if we go back to the structure of the protein, we see that there's only one ionizable amino acid in the vicinity of the substrate in the active site. The active site is very hydrophobic, right? But there's this tyrosine 459, which is close to the lipid substrate. And so we postulated this. We said, okay, we'll generate that first radical. That radical on that carbon should make that proton right there more acidic, all right? It's possible then that the conjugate base of tyrosine 459 could act as the proton acceptor to give what would be called a radical anion. And if you lose an electron from that radical anion, you would get this double bond. And we know that if you have a double bond, you can make a carbon-carbon bond via a radical mechanism, all right? And then in the second step, you would generate a second radical. That radical attacks the carbon, attacks the carbon having the double bond. That would put a radical at this carbon, which would abstract a hydrogen atom from tyrosine 459, leaving back a tyrosyl radical. And then that tyrosyl radical can get that electron back to re-give you the conjugate base of tyrosine 459 and your carbon-carbon bond. So that was our working hypothesis for how this reaction might take place. And so this is a very easy hypothesis to test. You just change tyrosine 459 to something that can't act as a general base. And so we changed it to phenylalanine as well as leucine. And in black, I'm showing you the activity from the wild type enzyme. And in green and red, I'm showing you the activity from each of the variants. And as you might see, even though the activity is decreased, it's still robust, right? The fact that you see any measurable activity is telling you that tyrosine 459 is not playing such a critical role in this reaction. We see the same thing. So that on the left was for the small macrocycle, but we see the same results for GDGT, which is the large macrocycle. In this case, both variants give you very close to wild type levels of activity. All right. So not wanting to let what we thought a good hypothesis go, we decided, well, maybe for some reason, we just can't see this intermediate that we're expecting, all right? And then we hypothesized, well, formation of each CSP3 bond requires 2 5' deoxyadenosyl radicals, right? So you need 2 molecules of SAM to form each carbon-carbon bond. And so we suggested that, well, if we only give the enzyme 1 molecule of SAM, we can only do half the reaction, which would then potentially leave us with this intermediate that we've been trying to observe, okay? And so we set our reactions up to look for this intermediate. We took our substrate, mixed it with the enzyme, purified the enzyme over a gel filtration column, and then we added all the other reaction components that we need for turnover, which is a low potential reductant and the 1 equivalent of S-adenosyl methionine that I told you about. And then on the left side, I'm showing you the results of this particular reaction. In red, I'm showing you that we're actually seeing the product being formed. In other words, not all of the SAM molecules are being distributed evenly, where only 1 is in each active site of the enzyme. Some active sites can gobble up more than 1 SAM molecule, and so that's why you're getting the full product. But in blue right here, that corresponds to a product that we didn't anticipate. And when we analyzed that by mass spec, shown right here, that product turned out to be a lipid wherein one tail had a sulfur or a thiol at the extreme end, okay? And we see that not only in a precursor to the small macrocycle, but we see that adduct as a precursor towards the large macrocycle shown right here. So this is GTGT, where you've made 1 carbon-carbon bond, but not the second. This is GTGT with that sulfur attached to one of the lipids. And this is our final product shown right here, all right? And so this is what we think is going on in this reaction, all right? So up here, this is our iron sulfur cluster that's bound in the radical SAM core domain of the protein. It's used to cleave SAM to generate your 5' deoxyadenosyl radical. That radical abstracts a hydrogen atom from the lipid tail. That gives you your lipid radical. And if you remember a few slides ago, I said, how do we stabilize this radical while we're generating the second radical? And it seems like the enzyme is stabilizing it by using that second iron sulfur cluster in the active site, the one in that C-terminal auxiliary cluster domain, as a protecting group, all right? So this radical then adds to that iron sulfur cluster to give this particular species shown right here, all right? And that's a relatively stable species. Now in the second half reaction, another molecule of SAM binds to the radical SAM core domain and is cleaved to generate a second radical, which abstracts a hydrogen atom on a second lipid tail. That substrate radical then attacks the carbon bound to the sulfur of an iron sulfur cluster. That liberates now your iron sulfur cluster intact. And at the same time, that gives you your carbon-carbon bond, all right? Now, why do we see this carbon-sulfur bond in our experiments? So this iron sulfur clusters are unstable in the presence of acid. So when we quench our reactions, the iron sulfur cluster breaks down because of the acid. However, carbon-sulfur bonds are stable, right? And so when the cluster breaks down, it leaves behind a trace of what was there before, and that's the carbon bound to the sulfur. Okay. And so this is what the crystal structure, again, of the enzyme looks like. And what I want to do is point out two phenylalanine residues in the structure that appeared to act as a gate to sort of guide the lipid into this sulfur of the iron sulfur cluster, shown right here. And this is a space filling model where you see that the phenylalanines, are acting as this guide so that you hit only the sulfur. The iron is protected by one of the phenylalanine residues. And so what we think is going on is this. So you rip off a hydrogen atom from the C-terminal tail of that particular lipid, and then you just rotate about this particular bond, and that places that radical right on that sulfur in the iron sulfur cluster. And that iron sulfur cluster acts as a protecting group. Now, as this rotates out of that pocket, either that lipid can rotate in to give you your small macrocycle, or one of these yellow lipids can rotate in that pocket, and that will give you the start to making your larger macrocycle. So that's what we think is going on. And so let me just leave you with a few conclusions for today's lecture. We have shown that MJ0619 is the elusive glycerol dibiphenylglycerol tetraether synthase. People have been looking for this for 40 or 50 years. We showed that MJ0619 for the first time in nature catalyzes a sp3 coupling between two completely inert carbons. The first radical intermediate is likely protected by addition to the auxiliary iron sulfur cluster. We have evidence for sulfur addition, but we need to do kinetics to show kinetic and chemical competence, showing that that intermediate forms and decays with rate constants that are consistent with the overall rate of catalysis. We think we've ruled out the possibility of an iron carbon bond intermediate based on the structure, but we have some spectroscopic experiments that are underway to try to show that unambiguously. What I didn't tell you about were these other cofactors that we have on the protein. We haven't figured out why we need this rubidoxin-like domain and why we need this N-terminal cluster domain. So those studies are ongoing in the laboratory. And then lastly, what are the motions of the lipids in the active site or from the membrane into the active site that allows for these bonds to be formed? So those are other things that we need to start looking at. And so lastly, these are the people who did the work. Cody is up there in the top left. He is the one who largely did almost everything. He was helped by Dave Iwig, research scientist in the lab with the native spray mass spectrometry. Bill Metcalf and his postdoc, Matteo Casu, sent us methanolsarcina acetovorans lipids to help us get started on this project. And my colleague, Amy Bohl, is a card-carrying X-ray crystallographer, unlike myself. And so she helped to mentor Cody in X-ray crystallography. And then lastly, we want to thank ALS and Argonne and funding from NIH and Howard Hughes. And thank you again for the invitation. I've had a great morning, and I'm looking forward to talking to people in the afternoon as well.
Thank you very much, Squire. That was a tour de force. So we've got time for a few questions. So I'll start with Anne.
That was lovely, Squire. I was intrigued by the temperature dependence of the reaction, possibly, or the
Right.
fact that the abundance of the macrocycle varies in archaeal membranes with temperature. Is it known whether that's at the level of the MJ0619 enzyme
I would
either?
love to answer that question. So, the question is so we showed that this enzyme gives two different products,
Right.
And so the question is, what governs that partition, right? And I would love it to be temperature. So at higher temperatures, you would get the GDGT, which is the large macrocycle. It's known that that's made at high temperatures. And then at lower temperatures, you would get the smaller ones. We're in the process of doing those experiments. The hang up has been that these molecules aren't easy to synthesize. And to do robust quantification, we need standards, to go in our mass spectrum, right? So what we did recently is that based on our structures, nothing is really interacting with the head lipids. I mean, these lipids are in the active site basically because of hydrophobic effects. So very little hydrogen bonding like that. So we hung off coumarin tags on the head groups of these lipids, and that will allow us to quantify them by using the UV absorption of the coumarin tag, as it's coming off HPLC. And so that's what we're doing
right now. Yeah, but that's exactly what we want to do, is see how temperature regulates what product is being made. I do.
Regulation of the enzyme itself?
Right, that's the other way it would most likely be done. You're right. But as an enzymologist, I'm hoping it's at the level of catalysis. That would be more interesting than a translational or transcriptional regulator.
One way or the other, either chemistry is going to rule or biology.
Exactly.
I am told that the clock is actually a little bit off.
Okay.
So we'll need to draw to this
Okay.
Q&A session to a close. There are as many people online as there are in the room, so that's another great testament to the power of the consequences of the pandemic.
Right.
To recognize your visit, Squire, I want to present you with this RCSB Protein Data Bank Yeti bottle that I hope you will use and treasure. These things will last forever. They're great quality. So
It's got to keep the people in my lab from trying to steal it.
well, that's
Everybody's always trying to put stuff up on the TV, so they're excited to ask that.
I think maybe we should start selling them at $100 a pop. So terrific talk. Thank you very much. And with the students, we'll migrate to the third floor for lunch. So thank you. Let's give Squire a hand.